When reading a scientific article, I basically try to get an idea of the details of the things I do understand. And the things that I don't understand, I try not to focus that much on because I might still be able to get the idea without knowing all the details. So I'm just going to model for you how I read this article and how I get an idea of the details of this article without knowing everything about it. So I'll go ahead and get started. Duchenne muscular dystrophy is among the most prevalent fatal genetic diseases occurring in 1 of 5,000 male births. Since I already know this, I just keep going. It results in muscle degeneration, loss of mobility, and premature fatality. DMD mutations are often deletions of one or more exons in the dystrophin gene that disrupt the reading frame of the gene and lead to a complete loss of functional gene dystrophin expression. And I already kind of know that there's a deletion in the DMD gene, and that causes the dystrophin not to work. In contrast, Becker muscular dystrophy is associated with much milder symptoms relative to DMD and is caused by internal in-frame deletions of the dystrophin gene, resulting in expression of a truncated but partially functional dystrophin protein. Because of the genetic nature of the disease, gene therapy is a promising option to treat DMD. However, the very large size of the dystrophin cDNA presents a challenge to gene delivery. Consequently, some therapeutic strategies aim to generate a BMD-like dystrophin. These approaches include the development of mini microdystrophin genes for delivery of by adeno-associated virus vectors, and oligonucleotide-mediated exon-skipping therapies designed to restore the reading frame of the transcripts 7-8. For example, removal of exon 51 can address 13% of DMD patient mutations, and exon skipping strategies could be extended to other regions of the gene to collectively treat 83% of DMD patients. In contrast, genome editing technologies can be used to directly correct disease-causing genetic mutations and may be a preferred approach for a single treatment to restore stable expression of a dystrophin protein that contains most of the normal structure and function and is also under physiologic control of the natural promoter. I'm going to stop there just because we went through a lot of text. And the gist of what I'm getting is that there's two things that they could do with the dystrophin gene, or there are two things happening with the dystrophin gene. In muscular dystrophy, the gene doesn't really work, and in Becker muscular dystrophy, the dystrophy kind of works. And they were saying that they could do gene therapy to try to get like maybe somebody who has DMD, Duchenne, to be more like a person with BMD, which has a functional dystrophin protein. It might not be like a perfect dystrophin protein, but it is functional, which means that it can work. Maybe it's a little shorter than normal, but it's long enough for it to attach the different parts of the muscle uh, fibers that need to be held together. And then I got an idea that the gene for DMD is very, very big, and that it's not as that easy to fix. I'm going to continue reading. In particular, the CRISPR-Cas9 genome editing system, which uses the Cas9 nuclease to cleave DNA sequences targeted by a single guide RNA, has recently created new possibilities for gene therapy by making precise genome modifications possible in cultured cells and in animal studies. And that means that they have done it in petri dishes and they have done it in animals. And I know specifically they had mentioned mice.